Okay, so in this segment of our discussion, we will try to know about different components of homologous recombination, and we would also like to discuss on the mechanism of homologous recombination in terms of molecular interactions. So, in a DNA, which is recombining the DNA, normally it was observed that there is a site called chi site. And this chi site has a kind of consensus sequence, which is shown over here, GCT, GGT, GG. And this chi site is target for some endonucleolytic cleavage. It is not a direct target, but the endonucleotide cleavage occurs somewhere near this chi site. So this is mediated by an enzyme complex called REC-BCD. REC stands for recombination. So it was observed that mutation on these sets of genes led to deficiency of recombination in some E. coli or in some bacteria. So that's why these are named as REC and then BCD, three different gene products which are active in recognizing the chi site and inducing the nick. This nick is important as it generates the invading strand. But DNA nick does not result immediately some invading strand. For that, the strand needs to interact with RECA. So what happens? This strand interacts with RECA and single strand binding proteins. So after this interaction, these strands invades the double strand, the target double helix, where it would recombine, and then it would scan for homology. So this scanning for homology is mediated by your REC-A, the protein, and um, these also mediate the strand exchange. So what would happen? Possibly this REC BCD would induce another nick in the second strand, and these two nicks would result into formation of a proper branch following sealing of these nicks. Okay, so here you can see, here you have a branch or holiday junction. Then this branch will migrate, and this migration would require two proteins which are RAV-A and RAV-B. Okay, RAV-B and RAV-A allows this branch migration, and with this branch migration, the strand exchange occurs. And after that, this holiday junction is resolved by a protein called RAV-C, which acts as an endonuclease. So here RAV-A and RAV-B acts as an helicase for branch migration, and each of these proteins has their specific role to play in inducing homologous, in mediating homologous recombination. Now the question is how these components were actually identified. So for that, we have to remember, we have to understand some experiment. So Clark and Anne Margulies, they actually performed some experiment where some egg plasmid was accepted by some E. coli, but that did not result in any colonies. So they were they were actually crossing some HFR strain with a recipient strain. And in normal conditions, the recipient strand is good, then that would allow some recombination. So the recipient strand was mutant in leucine and adenine synthesis. So following the uptake of the DNA from HF strain, these would become leucine plus and adenine plus. So they should be able to grow on minimal media. But they found that in the recipient strain, when the same cross was done with some mutant recipient strain, there was no colony on minimal media. And introspecting this event, they found out that the mutation is in a gene, and in that particular gene, if the gene is mutated, so recombination will not occur, and that's how these REC genes, REC gene was identified. Subsequently, it was found out that there are mutations in some other genes also, so those were named REC B, C, D, etc. So actually, the REC BCD they recognize a DNA, and then what it does, it actually it due to its in indigenous helicase activity, it can melt the DNA and it can move towards, it can move inward. And in this way, if it can find the chi site, then what it would do, it would induce a nick in the other strand, and thereby it would actually in through some exonuclease kind of activity, it would chew up the other strand, and it would allow the other strand to remain as a single strand. So this particular strand would be recognized by rake A, and this strand will invade the recombining DNA. So basically, this is how rake BCD acts to initiate homologous recombination. Now, as I mentioned, that chi site is very significant in terms of recombination. It was found out that for the genes that are closely linked to chi site, for those genes, the recombination frequency was much higher than the gene that are distant from the chi site. So here you can see the genes that are closely linked to chi site, they are 10 times more recombination prone than a gene which is some in the opposite direction of chi site. So here you can see it is 1x and it's quite distant. So it is far from chi site, and compared to that, a gene located nearby chi site, it has 10 times more recombination frequency. And this frequency gradually depletes even in the same direction if the gene is located distant from the chi site. So for A, it is 2.5 times, and here it is 10 times. So the recombination frequency gradually decreases if the distance increases from the chi site. So this is some observation which clearly indicates the significance of the chi sequence in homologous recombination. Now let us discuss about rake A a little bit. So this rake A, it is a 38 kilodalton monomer. It has several enzymatic activity, which includes ATPase activity, protease activity, and of course, both single-stranded and double-stranded DNA recognizing activity. So there are different domains you can see. There is a domain for ATP binding. There is a domain for double-stranded DNA binding, a domain for single-stranded DNA binding, this orange one. Then there is a part for double strand interaction. So a lot of different domains are there for double strand interaction, and specific domain for ATP binding, this purple one. And then a domain is there for hydrolase activity through which it can target some protein to degrade it. So different types of activities are there. It when it binds to DNA, it binds to a DNA with a ratio of 1:3. So that means in one RECA DNA binding pocket, there can be three nucleotides can be hosted. And this kind of interaction results in extension of the normal B helix of DNA to almost 1.5 times, that is 18.6 base pair per turn. When Reke forms a complex with DNA, also SSB single-strand binding proteins are found in that particular complex. So in Archaea, the ortholog of Reke is called RAD-A, and in eukaryotes, it's called RAD51 and DMC1. In T4 phage, the Reke ortholog is UVSX, so the names are different, but functions are more or less same. And when in the Reke SSB complex, the role of SSB is specifically to prevent secondary structure formation by the single-stranded DNA. Now, Reke is a multifunctional protein, as can be assumed by its, uh, uh, I mean, diverse functional domains. So here you can see that Reke can bind a single-stranded DNA, and this binding can induce its proteolytic activity, so it can trigger SOS response. Then Reke can, of course, bind a single-stranded DNA, and thereafter it can form complex with a double-stranded DNA scan for homology and mediate homologous recombination. So it's a second function, and also Reke can induce SOS mutagenesis by allowing UMUD, actually, which is forming polymerase 5 to replicate in a damaged DNA, and thereby inducing huge number of mutations. So there are diverse role of Reke, gene expression regulation, SOS response, as well as homologous recombination. Now, Reke assembly is quite interesting. So, as I mentioned you before that, a single Rake A, actually a single DNA turn can combine with three molecules of Rake. So initially, the first Rake molecule interacts with DNA, and subsequently the second and third molecule binds to DNA, and after that gradually the number increases. So there can be two possibilities. One is that following identification of the double-stranded DNA by Reke can assemble in a slower manner when this Reke multimer would progress towards the 5' end. So if this movement is 3' to 5', it would be slow. On the contrary, if this movement is 5' to 3', that means if the Rake multimer assembles in this way, so they can form the filament quite faster. Okay, so this is how this Rake can actually assemble on a DNA. Then identification of homology, there are different types of I mean mechanisms that has been hypothesized by several groups, that Reke can bind a DNA, and there is one primary binding site for Reke, and there is another secondary binding site for DNA. The DNA that binds at secondary binding site is tested for complementarity with the DNA that is bound to the primary binding site. And if the base pairing is stabilized, then only the strands are switched. So the DNA that is bound to the primary binding site, that is actually scrutinized for possible complementarity in the secondary binding site, and if it fits well, then only the strand exchange occurs. This was the initial hypothesis. Later on, in a very recent time, it was observed that this Reiki binding event is quite different. So if there is some DNA which is double-stranded and which is being targeted by Reiki for forming this D loop, what happens that this Reiki monomer bound to few nucleotides at specific sites, what they will do, this C-terminal end of Reiki will bind with the target double-stranded DNA, and subsequently this DNA, this Reiki will test for possible complementarity with the target DNA. If initially enough complementarity is found, then only this D loop will extend further, and this extension of this further extension of this D loop would actually mean that it would initiate recombination. So in terms of recombination, it is basically the first step is identification of the DNA. Then the second step is the D-loop formation initiation, and the third step is the synthesis of new DNA, the initiation of synthesis of new DNA by the homologous recombination mechanism. So this is how Reiki actually identifies the target site, and this is how it mediates the strand exchange. So in this scheme, this what is described in this figure A, the strand invasion part is basically mediated by Reiki. Next is the branch migration. So after this strand invasion and then formation of this holiday junction, as we mentioned earlier, that there is a need of migration of the holiday junction to a certain direction. This migration is mediated by uh, a complex called RAV-A and RAV-B. So RAV-B is an helicase I mentioned earlier, and RAV-A actually identifies the holiday junction. So what happens? This RAV-A RAV-B complex allows this holiday junction to migrate further, and this movement results in extension of the recombined strands. So, exchange of the recombined strands, further exchange of the recombined strands. So, if you see in this particular figure here, the actual binding stoichiometry of RAV A and RAV B, and those are shown. So RAV A can identify this holiday junction. It can bind there, and subsequently in both the sides to it can bind in the junction as a tetramer, and it can join with RAV B in the two sides. And similarly, mm, if you look into this complex from the top, it will look like that. So there is this Rave which is bound to this particular site, and the Raves that are wrapping around uh, the DNA, and thereafter it would start migration. So when it starts migrating, this is basically the view. Now, in the point of resolution, as we mentioned earlier, we need a endonuclease called RAF-C. So after the branch has migrated to a certain distance, RAF-C, the endonuclease would come. It would bind to the DNA as a dimer, and it can act on these single strands of the holiday junction to nick it and to resolve the DNA, the recombined DNA. So RAF-C, it binds with a stoichiometry of 2, So here you can see the dimer is bound to the holiday junction, and this binding can induce this endonuclease activity of RAPC, thereby it would resolve the complex. Now, a few terminologies, a few facts that we need to remember, that in any DNA, there is a preparation for uh, recombination, and this preparation we call as pre-synapsis. So in pre-synapsis, the DNA is combined with SSB and RAK, and subsequently the synapsis occurs. So when the synapsis occurs, this RAK bound DNA can form two types of joint. One is called paranemic joining, another one is called plectonemic joining. So in paranemic joining, this RAK bound DNA aligns with the target DNA, and it can start forming base pairing, and it can start forming the displacement loop. This is one of the possible uh, synapses. And alternatively, the DNA, while interacting with the DNA, the RAK DNA can directly displace one strand from the second DNA, and thereby it would form a single strand over here, and another strand will bound to the RAK bound DNA, and uh, uh, mm, to form DNA heteroduplex for recombination. So, as you can see here, in both of these paranemic or plectonemic paranemic and plectonemic joints, the actually the target DNA is snatched by the RAK bound DNA, and thereby they can form a double helix heteroduplex, and this can result in a displacement of the single-stranded DNA. So, this particular model actually supports what was proposed by Holiday model or by some other models, that RAK can actually mediate strand invasion and strand displacement. So these are a few snapshots for pre-synapses and post-synapses as was observed. So here it is the RAK binding to the single-stranded DNA that is shown, single-stranded linear or circular DNA. Then here it is shown that binding of SSB to and RAK to single-stranded DNA. So it's the electron image of that. And here this is if the DNA is bound to SSB alone, this looks like this. And if the DNA is bound with RAK and SSB, the DNA conformation looks like this. Then there are electron micrographic images of synapses. So here you can see these two DNA, they are aligning with one another like this. Here also they are forming some kind of joint. Maybe this is a plectonemic joint. And also here they can form some joint, some joint where we can see kind of strand invasion. So all of these are uh, basically snapshots obtained by electron microscopy. So here, at least the physical existence of such complexes was revealed. And then after there are some electromicrographic images of post-synapses. So that would mean that after those DNAs are joined, then some D loop will be formed. So here you can see this kind of looping is there. This is a loop. So this is what the D loop is. So even D loop could be visualized by electron microscopy. So all of these observations actually supported the basic model of recombination mediated by REC BCD, REC, then SSB, then RAV ABC. So this the function of all of those proteins cumulatively could be experimentally established. So for bacteria, there is a list for all the components which are involved in this whole process. So you can see there are different components involved in initiation, pre-synapses, then there are specific ATPases, then there are some proteins like some topoisomerases as well as some helicases that can directly allow the heteroduplex extension or branch migration. Then there are some endonucleases like RAPC, and also a group of other proteins which can mediate some auxiliary actions during this whole process. So a number of proteins are involved in recombination process in bacteria. Similarly, for most of these proteins, eukaryotic orthologs has been identified. So there are orthologs like MRE11, and then there are RAT50. So these proteins are involved in initiation of recombination. RPA is a single-strand binding protein in eukaryotes, followed by the proteins like RAD51, which is REC homolog, REC ortholog, and other proteins like BRCA1, BRCA2, and then XRCC in East it is RAD54. So all of these proteins are involved in presynapses. Then for heteroduplex extension, again RAD51 and few DNA polymerases like Pol delta, Pol eta, those are involved. Then for resolution, there are specific helicases, topoisomerases, and endonucleases. And then there are few factors which we call meiosis specific recombination factors, particularly the SPO11 and DMC1. DMC1 is the meiosis specific REC-A, that REC-A ortholog. So there are some factors which are meiosis specific recombination factors, and then initially the factors we named, those are mitotic recombination factors. So all of these factors are identified. So, and their functions has been experimentally validated. So these are the eukaryotic and prokaryotic components for recombination. Uh, then we have to know the components of double-strand break repair. So, as I mentioned before, that there can be double-strand breaks that are induced by specific condition. So those double strand breaks can be targeted by specific proteins like MRX or MRN, which can identify this double strand break. Then some endonuclease like SAE2, they can join these MRX in MRX in complexes, and they can mediate end trimming, so they can produce these 3' overhangs, and subsequently there can be some processing or resection of these 3' overhangs to finally form some invading strands with some helicase like DNA2, which can really start formation of single strands from these double strand breaks. So these are the components that are involved in initiating double strand break repair. For meiotic recombination, again, there is a double strand break formation as it is proposed. So SPO11, it binds to the double strand break. Subsequently, SPO11 is removed and the DNA is nicked by some exonucleases to form these overhangs, and then these overhangs can actually target their homologous DNA by using DMC1, the REC ortholog, and thereafter the crossover can occur. So these components are dedicated for meiotic recombination as it is proposed, and at the synaptic complex of some chromosomes undergoing through meiosis, these components can be identified. Some event and that is closely related to homologous recombination, uh, which are biologically relevant and which is also linked with some very interesting phenomena. So we have to understand those kind of events. One such event is mating type switching in yeast. There are other events also like expression of variable surface glycoproteins by some parasites. But for now, we would consider mating type switching in yeast. So in our next segment of discussion, we will discuss about mating type switching and some other biologically relevant phenomena associated with homologous recombination.